looking forward to and have been looking forward to tonight and talking to you a little bit about, what I've learned over these years. So I think Rob said I get about 3.5 hours with you, and then about 45 minutes of Q&A. So, no, I'm joking. It won't take that long to answer all your questions. But anyway,
You'd be surprised.
But this is a topic that is near and dear to my heart, but it's also a complex topic. For a number of reasons, not the least of which is that this has been a topic that has been debated in academic and non-academic circles for several centuries now. All the way back to and dating before 1859, when Charles Darwin published his Origin of Species. This topic has been debated since then. Okay, and because of that, then there's been an enormous amount of data that has been collected that has to be observed, has to be taken into account, and that has to be explained. And that then makes this field quite complex. Okay, over the last several decades, we've seen massive increases in our knowledge in my field, which is biochemistry and molecular biology. So it is complex, and especially when we start to answer the question that's said here, right? The origins. What are the origins of biological complexity? So, and it's an important question, obviously, for obvious reasons. When we think about the origins of biological complexity, and if we come to the right conclusion on what are the origins of biological complexity, what are our origins, when we come to the right conclusion on that, then we naturally are going to ask the next question is, why am I here? Okay, and if we come to the right conclusion on that, then our heart is usually ready to accept the gospel. Right? So this is a really important topic, and for all of us, but specifically it's near and dear to my heart, as I said. Now, I've said it's complex, and that's not to scare you, I'm just saying that it is. But at the same time, if you look at the title of my talk, what would a reasonable person conclude? I am going to try and present to you evidence tonight that doesn't require a bachelor's degree in biology or biochemistry or molecular biology. It doesn't require a master's degree. It certainly doesn't require a PhD in those fields to understand it. Okay, so I'm going to try and present to you the data in such a way that you can, first of all, access that data and then make a proper conclusion using that data. Okay, so that's my goal tonight, and I think that it can certainly be done. The talk I'm going to do really two parts. We're going to spend a small amount of time just looking at the biblical data. I think that's really important to think about it from a biblical standpoint. And so we'll address those first, the biblical considerations, specifically when confronting origins of biological complexity. And then we'll move into the other data, right? The outside of the Bible data, okay? And look at how that confirms what essentially the Bible affirms and teaches us. Okay, so that's where we're headed for the night. So let's go ahead and start here. I don't have a lot of verses, but this is one of my favorite. Okay, so Psalms 19, David the psalmist. Looking at. Well, I don't know if you looked at something quite like that, because that's probably a long exposure that then's been, enhanced through computer graphics and that type of thing. But it's beautiful, certainly. David, looking at nature surrounding him, penned this. He said, "The heavens declare the glory of God, and the sky above proclaims his handiwork. Day to day pours out speech, and night to night reveals knowledge. There is no speech, nor are there words whose voice is not heard. Their voice goes out throughout all the earth, and their words to the ends of the world." Right. So, what we're seeing here, and what David recognized, is that nature was speaking to him. Okay, and it was speaking to him about the existence of his God. Okay, and not only that, if you notice what he's saying is that nature speaks to all of humanity in the same way. There's not any language that nature doesn't speak, okay? And so the Bible tells us that God exists, okay? The Bible tells us that God created, but then nature tells us the same thing. Okay, Paul picking up on this exact same thing in Romans 1, very different context for this verse, but very similar concept. He says this, right? For the wrath of God is revealed from heaven against all ungodliness and unrighteousness of men who by their unrighteousness suppress the truth. Now here's why they suppress the truth. And here's what he's saying, that the reason that he knows that they're suppressing the truth. For what can be known about God is plain to them, because God has shown it to them. For his invisible attributes, namely his eternal power and divine nature, have been clearly perceived ever since the creation of the world in the things that have been made, so that they are without excuse. Right? So David very clearly here says that every human being that's ever lived on the face of the earth, whether they have access to special revelation, written word, or not, has access to natural revelation. That nature speaks to them, and they're going to be without excuse because of what God has penned in nature. Okay? So that's what the Bible tells us, right? And in summary, like I said, I've been pretty quick with this. Oh, I guess I shouldn't. I forgot about this slide. Okay, so that's okay. Let me rewind a little bit. So the Bible tells us that God created, right? And then he tells us that nature created, okay? Now, when we think about Paul, and we think about David, again, they were looking at something like this, okay? Now, we, in the 21st century, have, advantage in this way, in some ways, because we have tools that they didn't. The telescope for one, and the microscope for another, right? So while they could look up and see God, and they can look at the things around them, we can look down, we can look within, okay? And so what you're looking at here, this would be an image of a cell, actually several cells, okay, that has been stained by fluorescent dyes. And actually, this is a really beautiful image, but there's been stained by fluorescent dyes. And off to the right here, actually, let me see here, but this guy, this is a big cell right here, okay? What you're seeing here, that blue portion of the cell, that's the nucleus, okay? And then the green and the red are the cytoskeletal elements of the cell, right? So human beings have a skeleton that's made of bones. Well, cells have a skeleton as well. We call it a cytoskeleton, and it's made up of proteins that make up what's called macrotubules and actin filaments. And so the actin filaments and the macrotubules are shown here in green. Okay, and all I want to say is this: that we have now the capacity not only to look into the stellars and the heavens, but we have now the ability to peer into the cell, see the complexities, and see that nature is speaking to us, okay, and that we should then, like Paul admonishes us, we should be able to then draw conclusions from that. And if we're drawing the wrong conclusion, as Paul says here in Romans 8, or Romans 1:18, he's saying that you're suppressing the truth in your righteousness, right? Jesus says something similar in John 3, where he says that men love darkness. Okay, after the famous John 3:16, right, he goes on and he describes that the light has come into the world, but men reject the light because they love darkness, because the light exposes their deeds. Okay, but it's plain to them. It's clear to them, because there's not going to be anyone that stands before God that says, "Yeah, but," and then he's going to go, "Oh yeah, I forgot about that. You're right. You get the free pass." Okay, that's not gonna happen. Okay, all right. So, in summary, okay, the Bible tells us one, that God created, but then the Bible tells us that then nature tells us that God created. Okay, and so what we want to do now, what I want to jump into for the rest of my talk, is looking at that data from nature, specifically in a field that I'm very familiar with, and try and help you to see what I see is so clearly written. Okay, now let's do this. Okay, so we're thinking here about the origins of biological complexity. I'm a teacher primarily. Okay, so I don't hold a research position at Colorado State University. I'm really more there from an educational standpoint. And I know this, that you usually want to define your terms. Okay, so what exactly do I mean by biological complexity? So this is this is a quote from an encyclopedia. So there's an encyclopedic definition of biological complexity. It says this: refers to a measure, biological complexity refers to a measure of the complication of a biological organism that is directly related to that organism's ability to successfully function in a complex environment. I'm just sitting there saying, I don't know if that helped me out, but thank you for that. What are we talking about here? Okay, let me try and unpack this definition for us all. Okay, so I actually start my class off, okay, one of my classes, in this way. Okay, so I say, if we take in a reductionist view of life, or a reductionist view of a cell. Now, what do I mean by reductionist view? I don't say this in my class, but what I mean by that is that I'm saying that we're only thinking about the physical aspects of life. Okay, as Christians, we believe that we're dualist at the middle. Okay, we have an immaterial and a material being. Okay, so when I say reductionist view, we're thinking about just the material things of the cell. We can boil it down to this. Okay, a cell is made up of parts. Okay, all cells are made up of parts. Okay, and understand that you have trillions of cells that make up you. Okay, but in each of those cells there are these parts. Now, what are those parts? Well, there are four main, what we call macromolecules. The word macro meaning big. Okay, so macromolecule means a big molecule. And there are four classes of those. And you probably have heard of these. Proteins. And yeah, I think you've heard of proteins. Especially with our diets these days, right? But proteins are one of them. Lipids. Yeah, I think I know lipids. Carbohydrates. Okay, and then finally nucleic acids. And you say, I've heard of those first three. What is nucleic acids? DNA. You know, oh, okay, I know what DNA is. DNA stands for deoxyribose nucleic acid. Okay, so those are our four main big parts of the cell, big molecules in the cell. Okay, nucleic acids also has RNA as well. We can talk a little bit about that as well. Okay, but then there are all these other molecules that are small molecules. Okay, what we call metabolites, or just small molecules, and there's thousands of those in the cell. Okay, so when we think about the cell, it's got parts, molecules, lots of molecules, big ones, small ones, okay, different classes of them. Now, those parts then come together to carry out processes. Okay, so yeah, you've probably heard of processes like cellular growth and division. Okay, is a process that occurs in a cell. Okay, it's how cells replicate. Okay, metabolism is a process that occurs within a cell. Okay, there's all sorts of these processes that biologists and biochemists alike have discovered, and we've defined, and we've talked about them. Okay, but the parts are going to come together, and this is really where the chemistry of biochemistry comes in, is that these parts come together and they do things. Okay, these processes. Now, those processes go in certain directions. Okay, cells grow and then they divide. Metabolism takes nutrients, breaks them down to make to store the energy from them in the molecule called ATP. But they go towards the breakdown of those things. Okay, so they're going in certain directions. And the reason that they're going in certain directions is for these bottom two, right? There are these certain driving forces, okay? Thermodynamics, if you're familiar with chemistry, you've talked about thermodynamics, okay? But these are the driving forces, the energetic of the cell, the reason that they go in a particular direction, okay? The driving forces, and then there's all sorts of information. Okay, those nucleic acids are the blueprint of the cell, and they're directing the cell and what it should make and what it should not make, and those kinds of things, okay? So that's a cell, okay? It's got parts. Those parts come together to do processes, and those processes go in certain directions because they're driving forces, and they have information to it. Now, if you were in my class, I'd say, okay, for the rest of the semester, that's what we're talking about. Okay, and we'd spend the next 15 weeks, 4 days a week, 1 hour a day, talking about that. So, like I said, 3 hours is short compared to what I just put my students through this last semester. Okay, and you're not going to get a test at the end of this, so you just count yourself lucky. Okay, now if you're wondering, you're still sitting there saying, help me out a little bit. How about we go here? And you're like, oh yeah, okay, so that's a GMC Sierra. Okay, a nice GMC Sierra. I drive a 2006 Toyota Tundra. I like my truck, okay? But it's not this truck, okay? That's for sure, okay? But when we think about it, we can just think about this is a really, 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 really big cell, Okay, why do we say that? Because it's got parts, right? Lots of parts. In fact, I got into Google and I searched how many parts are in a GMC Sierra, and it said lots. It's very helpful. And we'll just leave it at that. It is true. There's a lot of parts on this thing, right? So, but it's got parts, but then it's got processes, right? There's all sorts of processes that are going on in this GMC Sierra. The engine, right? When you turn it on, there's tons of processes that are going on. But you roll down your window, you got air conditioning, you got heating, you got so many different processes that are going on in the GMC Sierra. Okay, and of course those processes need the parts to go, right? Now, when we think about then, well, how are they all directed? Well, energy, right? You need energy, you need driving forces. We put gasoline in this pickup, okay? We put a big battery in this pickup, right? That battery and the gasoline and all that is going to send the pickup in a certain direction, the process is in a certain direction, and oh yeah, there's a ton of information in it. Okay, in fact, these new vehicles, right, they have like an entire iPad sitting in front of you, right? And there's entire software programs that are written to run these things. So there's tons of information, right? So that is essentially a very large picture of what I mean by a cell and biological complexity. So we can just come back to here. Let me go back this direction. Okay, let me redefine here for you, or let me reread this for you, and think about this as automobile complexity. This refers to a measure of the complication of an automobile, right, that is directly related to that automobile's ability to successfully function in a complex environment. You think, oh, so you mean that these have to be complex because they have to do complex things? Exactly. That's what we mean. They have lots of parts, they have lots of things that carry out these processes to function in a complex way. And that's what we mean by biological complexity. And I'm gonna be honest with you, if you Googled how many parts are in a cell, you're gonna say more than the GMC Sierra. Okay, truly more, okay, in each cell. Okay, so it's very complex, okay, and our question that stands before us is, well, what then is the origins of that complexity? The parts, the processes, the driving forces, and the information. Okay, that's what the question is before us. And there are two then hypotheses for this, okay? Broad hypotheses. One, an undirected material process brought all of that complexity into existence. Right? This would be the chance hypothesis. We are a fortunate accident, okay? And biological organisms are a fortunate accident. Some people might think, well, I think it's unfortunate. Okay, well, I think it's fortunate. I enjoy living, okay? I enjoy the life that God's given me. Okay, Neo-Darwinian theory would be the prevailing mechanism for how that all came into be, right? So neo meaning new, Darwinian, okay, theory, okay? So we could, spend hours defining that, but we just say this, that this is essentially the powers of natural selection acting upon random mutation, genetic variation to produce biological innovation and complexity, right? That's the prevailing theory, at least in this top, right, undirected material process. The other one would be this, a material process, but directed by an intelligent being. Okay, that's our other hypothesis, right? So now, is that one that I want to spend some time talking about, because is that one that suggests that, okay, nature speaks to us, because there's an intelligence, an engineer behind it, okay? And that's what we're going to talk about. So let's think about it this way, okay? We're going to try and look at both of these questions tonight, okay? We'll spend, hopefully we get to number two, because number two is pretty cool, but we'll first talk about this first one. And the question is, are there features of a biological organism that exhibit the marks of intelligence? Okay, so in other words, can we look at a cell and say, that's because there's a brain behind it, okay? And so we're gonna look at those features, we're gonna try and look at those features. The second one then is this, and this one can get very complex, but the second one is this: are there features of biological organism that current neo-Darwinian theories of evolution cannot explain? In other words, these are what we know is the power of Darwinian, the powers that would be found in neo-Darwinian theories. Could they possibly explain it, or are they going to fall short? Okay, so we'll try and look at both of these. Okay? All right, so let's look at this first one. Are there features of biological organisms that exhibit the marks of intelligence? Now, I'm going to take you down this road, right? Okay? And so here's what we want to do. I want you to imagine that you just walked into a room, and in that room is a lotto ball machine. Okay? And the lotto ball machine has a very large hopper that has lots of balls in it. Okay? Now, unlike lotto ball machines that have numbers on them, we're going to say that this lotto ball machine the balls have letters on them. Okay? And you walk into that room and you see, okay, in this little portion of it, right here, right? You see a whole bunch of balls lined up, 150 balls lined up, okay? And they have letters, and see that the sequence of letters that's found there is this one, okay? It's a huge sequence of letters. Okay? Now, you think to yourself, okay, I see that. Now, just, you can even answer this question. Would you attribute that sequence of letters to intelligence? I mean, you might just say the machine. Don't think about the machine. Don't think about anything else. Just the sequence of letters. Would you look at that and say an intelligent mind brought that together? Or could anything have pushed a button to bring about that sequence? Most of you are saying, I'm pretty sure that, my dog could have done that. Okay? So, not that your dog isn't smart. Well, some of them are, right? Some of them are not so smart. Okay, I had dinner about that. I had a conversation about that over dinner, but we won't get into that. Okay, so you can look at this, and you can probably think that it's safe to say that an intelligent mind is not behind that sequence of letters because it appears to be completely random. Okay. Now, let me say this, okay? If you were to take all the balls out, put them back in the hopper, and you push the button again, would you get that sequence again?
No.
No, you would not get that sequence again. It is extraordinarily improbable that you would get that sequence again, okay? In fact, some of you are already doing the calculations, okay? This is the calculation, right? So the chances of getting that sequence again is 1 in 26 raised to the 150th power. Some of you are like, I hate math. Why did you just do that to me? Like, I hate it. Like, what are you talking about? Okay, so let's think about this. The first letter in our sequence is L, okay? You have a 1 in 26 chance, because there's 26 letters in the human alphabet, you have a 1 in 26 chance of getting an L on the first push of the button. You think, I got you. I think I can do it, okay? The next one, if you wanted to get LX, right, because the next one's X, you'd have a 1 in 26 chance to get the L, and then a 1 in 26 chance to get the X. You'd have 1 in 26 times 1 in 26 chance of getting LX, right? Or we just say 26 raised to the power of 2. Okay, 1 in 26 raised to the power of 2. Okay, you get the third, you get the fourth, you get the idea. I put 150, okay, as the exponent because there's 150 letters in our string of characters here. You think, okay, I got you. 1 in 26 to the raised to the power of 150. Now I'm a scientist, and I have been trained to think in scientific notation. It's just like, I can't, like 26 raised to the 150 doesn't mean much to me, so I think about it in this way, okay? I put it in scientific notation. This would actually be a 1 in 10 raised to the 212 power, okay? Some of you are like, that's a really big number, I think. Yeah, it's 1 with 212 zeros behind it. It's a big, big number. Okay, now, let's think a little bit about how big this number is. Okay, and don't worry, I have a point with all this. I'll get there. Like I said, I got 3 hours, so we're going to be fine with it. Okay, so, but let's think about it this way. 1,000,000 in scientific notation is 10 raised to the 6. Okay, now the thing that we don't think about with exponents, I was thinking about this. If you think about exponents like 10 raised to the 1, the first power is 10. We can count to 10 right now. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Done. That's not a very big number. If I go to another exponent, 10 to the second, that's 100. I could count to 100 right now. I go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and then I have to do that 10 more times. It takes a little bit longer. Okay, I'm not going to do it. If I go 10 to the third, that's 1,000. Now I have to count to 100 10 times. If I go 10 to the fourth, that's 10,000. I have to count to 1,000 10 times. right? And you can see how long it's going to take me to start counting. If I have to go to 10 to the 5th, okay, I'm going to have to count now to, what is it, 10,000 10 times. You get where I'm going with this, right? We go 10 to the 6th, we're at a million, okay? A billion's 10 to the 9th. A trillion is 10 to the 12th. Trillion is a huge, huge number. I have to count to a billion, right, 10 times to get to a trillion, okay? So yeah, it's a big number, okay? $35 trillion represents our national debt. Ouch. At a rate of $10,000 a second, it'll take us 100 years to pay that off, or more than 100 years to pay that off. $10,000. Think about how big that number is, okay? And now, our odds of getting this sequence is 1 in 10 to the 212th, right? 200 orders of magnitude larger than a trillion, okay? It is an enormous number, inconceivably large. In fact, and don't ask me how they know this, because I'm not an astrophysicist, but astrophysicists say we have estimated the number of atoms in the universe, and it's 10 to the 80th. The number of atoms in the universe, which means that we would need 2.5 universes of atoms to get to this 10 to the 212th number. It's big. So yeah, you're not going to get that sequence again, pushing it. It's just not going to happen. But you keep saying, you're thinking, where is he going to this? That's not the sign of intelligence. Just because it's extraordinarily what we call fantastically improbable to get that, does not mean it's the sign of intelligence. What about this one? And you're like, you should have used a larger font. I should have used a larger font, I'm sorry. I didn't realize how big this was going to be. Let me help people out. This one. Okay, that's the same sequence. If you walked into this room, okay, and you saw this on the lotto ball machine, would you think that that's the sign of intelligence? That sequence of letters? Similarly, if I see what I'm seeing, I think I would. Okay, this is a quotation from Romans 1. Right, let me read it. What can be known about God is plain to them, for his invisible attributes, namely his eternal power and divine nature, have been clearly perceived ever since the creation of the world. That, okay, if you walked in and you saw that in a lotto ball machine, you would automatically think that an intelligent mind put those balls there. You would not think it was a random process at all. But that's 150 characters. You have the same probability of getting that as you do this, right? One of them has the marks of intelligence, another one has no marks of intelligence. So what's the difference? The difference is that not only is this what we call a complex string of characters, complex meaning it's extraordinarily improbable that you would get it through a random process, but it also is very specific. And it's specific to the English language, right? It follows the rules of English spelling and English grammar rules, right? So this is what we call specified complexity, and no one in their right mind should look at this and think an intelligent mind is not behind that. No one, and no one would. There wouldn't be a person in the world that's ever lived at that would think that's not the sign of intelligence. So we have within us the capacity to recognize something that has the marks of intelligence, right? Let me show you this. What about that? Marks of intelligence? Yes or no?
Yes.
Oh, absolutely, 100%. I can't talk about probabilities. What are the probabilities of getting that? We're not going to talk about that. But when you look at this, there would be entire manuals of the company that made this that have engineering specifications on this, right? This is a piston that will be found in an engine, right? And it have to have engineering specifications because it has to fit within a particular cylinder and, right, I mean, all sorts of things, okay? So you would look at this structure and think, yeah, absolutely, 100% that has the marks of intelligence. This would also be what we call specified complexity, okay? It's a form that has a very specific structure and a very specific structure that is related to a very specific function. And without that specific structure, it will lose that function. Okay? Now, we can then conclude this little portion of our talk here and say that it is uniform human experience tells us that things that exhibit specified complexity are the result of intelligent beings. And in the sciences, we make this conclusion all the time, right? Think about the archaeologist. When the archaeologist goes on a dig, if they find a tool in that dig, or they find an inscription on a cave wall, or whatever, their first thought is, it's not even a thought, they assume that an intelligent being brought it into existence, right? Now, if they had encountered a piston in their dig, they might think, wow, this was a really advanced society, or crap, someone contaminated my dig, right? One of the two, right? They would think one of those, okay? But archaeologists make this they make this decision all the time. They automatically assume that intelligence is behind it, and then they try and figure out, well, what does this tell us about the society that made this tool or had this inscription? Okay. Now, if they were to say no, we need to exclude all other possible explanations besides a naturalistic explanation for the existence of their tool, then they would exclude the right answer, correct? Mm-hmm. Right? They would have to try and figure out, well, why is this fork here? I think wind, I think time, I think erosion, they're going to come to the wrong answer on that, okay? So they do it all the time. For some reason in biology, they won't let you do it, okay? Okay, so, but the point of this is that we have in the sciences, and we have in ourselves the capacity to recognize when something has been put in place by an intelligent being. And we can just define that as exhibiting specified complexity, okay? All right, now, back to our question. Are there features of biological organism that exhibit then specified complexity? Okay, so I'm gonna say yes. I'm gonna try and now draw on your intuition, right? And again, think about my talk, right? The origin, the title of my talk, Origins of Biological Complexity, what would a reasonable person conclude, right? Go back to our sequence of letters. A reasonable person would conclude that a quotation from Romans is the result of not a natural process, but of an intelligent being, okay? All right, so let's do this. I want to look at, because I'm a biochemist, I want to look at one of our macromolecules. Remember, these are the big molecules that are part of the parts that make up the cell, okay? We want to look at proteins, okay? And if you think about like the pickup, go back to our pickup, the proteins are like the parts in an engine. I mean, they are there and they're doing lots of stuff, and there's tons of them in a cell, okay? Now, a protein. Okay, great. So a protein is this. It's a complex macromolecule, okay, comprised of amino acids linked together in a specific arrangement to carry out a particular structure. Oh, I'm sorry, not structure, but particular function. So it's a complex macromolecule comprised of amino acids linked together in a specific arrangement to carry out a particular function. You think? Help me. Okay, I'm gonna help you. All right, so let's go here. An amino acid. Okay, so when we think about a protein, proteins are made up of building blocks, and those building blocks we call amino acids. Just like a Lego, right? If you think about a structure of Lego, my kids love Legos, okay, in particular one Legos, he's sitting right over there. Okay, but so Legos have building blocks, and we put them together to build these really cool structures, right? So do proteins. They have a building block, and that building block is an amino acid, and you're looking at that, an amino acid right there, and you're thinking, oh, so I have these little spheres in my body? Not well, kind of, yeah. I mean, so these represent atoms, okay? So an amino acid is made up of this blue one, which is nitrogen, and then this white one, which is hydrogen, and then these green ones are carbon, okay? And then this red one is an oxygen, okay? And then this gray one, well, it's more than one, actually, atom is what we call a functional group, okay? Or an R group, okay? And it's very specific for what amino acids we find. Okay, so it turns out that when we think about proteins are made up of 20 different amino acids, the building blocks. Okay, what differentiates amino acids are this one right here. Okay, this appendage that we call an R group. It has a very specific structure and has very specific chemical properties. Some of them are like really love water. okay, what we call them hydrophilic, okay? Some of them really hate water. We call them hydrophilic, okay? And they try and get away from water, okay? You think to yourself, I didn't think atoms had the capacity to love or hate. They don't, it's just, euphemisms, okay? I don't have that, okay, but that's the idea. Now, when we think about a protein then, we link these amino acids, these building blocks together, right? So if you think about this, look at this structure here, we see this structure right here, right? So that's one of the amino acids in this. Okay, then we go to this one, we've taken that structure and now we've turned it upside down, okay, but it's right here. There's a second amino acid right here, and then there's a third amino acid right here, right? And hopefully you can kind of see, I think I see the pattern here, the repeating unit that we have in this, okay? And so this would be a protein, a very small protein. It's only made up of 3 amino acids, okay, but it's in this chain, okay? And then there's these R groups that are different for each of them. Okay, the 20 different amino acids. All right. An average size protein is about 300 amino acids long. Okay? And so let's go ahead and look at a protein. And you say, oh gosh, lots of information just came up on the slide. Yeah, I know. Okay. So let me try and take you through this. Right here, all these letters, you think that just is a lot of letters. It is a lot of letters. It's 260 letters. Each of those letters represents one amino acid in a chain of amino acids. Okay, we call this, as scientists, we call this the primary structure. Now, we could go back, it would actually look like this, okay, and these little R groups would be different for each of them, okay, and there would be 260 of them that make this long chain that looked like something like this, but we just put it in letter form, okay, right, and we call that the primary structure. And this happens to be the primary structure of a protein called chymotrypsin. And you say, means nothing to me. I mean, really, chymotrypsin means nothing to me. So chymotrypsin is an enzyme. It's found in your stomach, okay? Well, it's actually found in your GI tract, more in your intestines, okay? And we're going to talk about what it does, but it's a digestive enzyme, okay? Now, here's the important thing about this. This sequence, specific arrangement of amino acids, leads to this. Okay, this is a 3-dimensional structure. Think back to these appendages, these R groups that I talked about that are different. Go back here. Right, these guys. Some of these love water, some of them don't love water. And they have other chemical properties that we don't need to talk about. Some of them are acidic, some of them are basic, all sorts of things. Okay, but the idea here would be this, that these in that chain, you think about it as a chain, and then you put water around it. Some of them are going to start to run towards each other in the chain, and some of them are going to run right to get away from the water, and some of them are going to run toward the water. Okay, but they're all linked to each other. And what they're going to do is they're going to fold up into a glob, okay, or a 3-dimensional sphere. We actually call them globular proteins. Literally, that's what we call them, globular proteins. Okay, and they have these tight, compact, 3-dimensional shapes that are kind of a glob or a sphere. Okay? And that's essentially what you're looking at. So this means this. Okay? Now you think, well, what's that over there? Well, let me go ahead and describe this. So this here is what we call the cartoon version or the cartoon view of a protein. This is how we present proteins. Okay? And cartoons, right? Mickey Mouse is a cartoon. Mickey Mouse has the features of a mouse. But then Mickey Mouse has the features that are not mouse-like, correct? Right? That's what this is. This is a depiction of a protein that is like a cartoon. It has features that look like a protein, but then there are features that do not look like a protein. Okay? But it's a nice way to look at it. Really what the protein looks like is probably something more like this. This kind of glob that has lots of this like protrusions, the very contoured surface. Okay? Now, when we look at this though, let me go ahead and highlight this for us. Okay, you see here, right in here, you see these kind of teal colored right there where I'm hovering over. And there's these like ball and stick things right here. Okay, those are appendages of these amino acids that I've highlighted here, H, which means histidine, D, which means aspartic acid, and then S, which means serine. Those are all right here. Now think about that in terms of the chain. Look at how far apart they are from one another in the chain, but look at how close they are to each other in the protein. This thing is folded up into a structure. Okay? Now the placement of those is extraordinarily specific. They have to be placed there for this protein to do its job. And you think, what's its job? Here's its job, okay? It does this. And you say, oh, that's what its job is. Thank you, so helpful. What in the world is that? Okay, that is a chemical equation. You're thinking, oh man, nightmares. Okay, well maybe. Okay, so let me try and help you out here. So this protein, chymotrypsin, like I said, it's in your digestive, or it's in your GI tract, it's a digestive enzyme. What its job is to do is when you eat a protein-rich meal, okay, which you should do, protein-rich meals are important, okay, you need enzymes that take that protein and break it up into its individual amino acids, because your body doesn't have the capacity to absorb an entire protein, they're too big. So we have to break them down into their individual amino acids, okay? So what you're seeing here is one amino acid here, one amino acid here, and then this arrow, if we go in this direction, those amino acids come apart. And it's called a hydrolysis reaction because they need water to do it, okay? Now that reaction, if you have just this and water, it'll take, well, it has a half-life of 450 years. So if you have 10 grams of this protein, that means it'll be 450 years when it's surrounded by water before it's 5 grams of individual amino acids. Okay, and 5 grams still of the protein. That's its half-life, okay? Think about the last protein bar you ate. If it took you 450 years to digest that thing, you'd be older than you are, right? I mean, yeah, right? Your body cannot survive at that rate. So what it needs is an enzyme. An enzyme catalyzes the reaction, and this right here, chymotrypsin, will take the rate of that, which takes 450 years, down to less than a second. It'll do this in millisecond timescales, okay? In fact, there are some enzymes that can catalyze, not this enzyme, but there are some enzymes that can catalyze over 1 billion reactions a second. 1 billion reactions a second, okay? Every single one of those enzymes that I'm talking about has a specific amino acid structure and a specific structure that comes with it. Okay, so go back to what I was talking to you about specified complexity. We use 2 different things to illustrate this. Think about the piston, okay? The piston had a very specific structure that had a very specific function. That is what this is, okay? So this is a very specific structure with a very specific function. If you disrupt this structure, then you will disrupt its function. It will not be able to catalyze this reaction, making it, go from 450 years to less than a second to do. It just won't be able to do it. Okay. The other thing is this, that structure, okay, is coded by this sequence of amino acids. Okay. So go back to what we were talking about with our lotto ball machine. We had a very specific sequence of letters. Here we have a very specific sequence of chemicals, right? Amino acids that dictate this, okay? In fact, this is fairly intolerant to changes, okay, mutations. If we start to change out amino acids here, okay, in this structure, then we will lose this structure here and its ability to function. Okay, just like the English language. There are letters that we can start to change and you start to lose meaning in the sentences that are written. Okay. Now, in this case, there are some of these amino acids that we could change and have no effect. Okay, so we could change potential. I don't know which ones, but like you could change this one, let's say, to whatever, one of the other 20, nothing would happen. No visible change in structure, no visible change in function. Okay, but there would be other ones, like this one here, okay, and this one here, and this one here. If we change those at all, you would lose the entire function. It would be an unfunctional protein, okay? And the more we change in this, the more likely we're to lose the structure and the function of this, right? So what I'm trying to say to you is that this is specified complexity, okay? It has a digital sequence that makes up a very specific structure with a very specific function. What would a reasonable person conclude if they encountered this? Right? That's the question. is this a feature of a biological organism that exhibits an intelligent mind behind it? I would say yes. Okay, now, the rabbit hole goes deeper. You think, yes, I want to do it. Yes, let's go deeper. Okay, let's do this. Okay. When you think about this, let's come back to this first before we get to that next slide. If what I'm telling you is true, and it is, okay, that is backed by scientific data. Okay, so I mean, there's a lot. So you have to start to think to yourself, well, how does the cell make sure that this sequence is made every time when it wants to make this protein? Okay, and it's going to make thousands of these. Your body, this protein is actually made in the pancreas. Okay, your pancreas is going to turn these things out all the time, 24 hours a day. Okay, thousands and thousands and thousands, if not millions, if not tens of millions of copies in your lifetime. And it has to make them the same way every time, because if it doesn't, then it'll be mutated and you won't like your protein-rich meal anymore. Okay, it'll be an ouch. Okay, so it's very specific. So how does it do that? There has to be some process to ensure that it can do this, right? Some of the engineers in the crowd are like, yeah, processes. I like processes. Let's talk about processes. Okay, so let's do this. Think about this, okay? Let's go back to our Lotta ball machine. All right, now we got our Lotta ball machine and we have this sequence of letters. You walk in and you say, not the marks of intelligence. But then you get closer to your Lotta ball machine and you see that on the Lotta ball machine there is a piece of paper that gives a description of how you can decode the information here to the information here, right? So there's a code. And you do that. And the code says, every time you see an L, put a W. Every time you see an X, put an H. And you do it, and you think, and you come from here to here, and you think, wow, someone spent way too much time on this. Because they just made this code, right? But that's what they did, right? And you would think to yourself, that's the sign of intelligence. Because we know that codes and the processes that decode them have to be the signs of intelligence, right? Okay. Let's do this. Let's think about this now in a biological sense. In a biological sense, our codes, okay, our proteins that I was just talking about are coded for in the DNA. Okay, so that's what you're looking at here. This is the DNA. And you and I, in every single one of our cells, there are 3 meters of DNA in every single one of your cells. And there are trillions of cells. 3 meters, 10 feet. Okay, that's a lot of DNA. 3 billion what we call bases in one cell, all arranged in a very specific pattern, okay? That's our genetic information. Now, DNA is like a protein in some ways, okay? It's made up of little chemical units, building blocks. They're not amino acids though, they're nucleotides, okay? I'm not gonna show you the structure of a nucleotide, but there are 5 of them, okay? There's G and C, there is T and A, and then if you're talking about RNA, there's U. Okay, so there's 5 of them, okay? Unlike proteins that have 20, okay? Amino acids, these have 5 different nucleotides, okay? And they come in very specific arrangements, very specific sequences in our DNA, all right? Now, 3 billion bases, okay? Now, if you think about this, let's look at this portion of this. Okay? This represents a gene, and we'll just call it the chymotrypsin gene. Okay, the chymotrypsin gene is going to encode the information to make this, to do this, okay? So we have it in a DNA form right here, okay, the gene. Now, the cell has to have a decoding process. That's the code, it has to have a decoding process. And that decoding process is going to start here. It knows exactly where to start the decoding process. It's going to start right here, and it's going to end right here. So there are molecular signals in our cells that tell some proteins called RNA polymerase, it's a huge protein, significantly larger than the one that we looked at, to start making a copy of this DNA in a process called transcription. Oops, okay? And that'll make then what we call RNA, or what we call pre-mRNA. So it's taken DNA and made RNA from it, okay? But it's essentially the same language. Okay, we're still using the language of DNA. It's still A's. It's still, instead of T's, it's now U's. It's G's and C's. Okay, okay, and that's this information. But this information has the information eventually to get down here to the protein, okay? But you see here, I've got these little sections like this one here, this section here, this section here. When this process occurs, there are sections of it that are not quite so useful. Okay, they're useful, but they're not useful for getting essentially down here. So this has to be removed through another protein complex called a spliceosome. We have to take those out, okay? And they have to be taken out in very specific ways. If they're taken out in the wrong ways, then we'll ruin the protein, okay? But fortunately, the spliceosome gets the molecular signals to know where to do this, okay? And then finally, it's got mRNA, okay? But still coded, still can't see the protein. Okay, so what has to happen? What has to go through another process called translation, and that takes another huge molecule called the ribosome to do this, and the ribosome knows exactly where to start and exactly where to end to decode the information that essentially came all the way from the DNA. You think, you lost me. Okay, let me just put it this way. This is a decoding process, and it's extraordinarily more complex than the process that I talked about right here, where all I said was L means W, right? It is extraordinarily complex. It requires other machines in the cell, and it has to pick out of 3 billion bases the exact right ones and start in the exact right place and end in the exact right place, or the whole process will be mucked up. And you will be mucked up as a result, okay? And you won't like it. Some of you are like, did he just tell me I'm going to be mucked up? No, I'm just saying that that's what's going to happen, right? I mean, these genetic diseases muck us up, right? I mean, it's hard. Cancer is a genetic disease, okay? Not a laughing matter, quite honestly, but anyway. But when we look at this, let me say this. We've got mRNA, right, and we get the protein. What's the decode, right? How do we decode that? Well, we have this. It's called the genetic code. And we have took decades to break this. But as a biochemist, we broke what's called the genetic code, and we now present it to students as this. And we say, this is genetic code. And we say that there are in mRNA, there are 3 codons that will translate to an amino acid, right? So the first amino acid in this is an M, methionine. That is, in RNA language, that's AUG. Okay? AUG means or equals M. Okay, so in the mRNA, we'd see AUG, the ribosome, this protein that is the decoder, knows it's a protein RNA complex. But anyway, AUG, that ribosome knows that AUG equals M, so it knows to take the amino acid M and start our chain of protein. Okay, the next code, L. The next amino acid is an L. Okay, so L, okay, is CUU. That's the next code, what we call codon. And it's just going to go through this, right? There would be a code, a very specific codon for each amino acid here to ensure, and this is our process, to ensure that this sequence of amino acids is made every time the cell needs to make chymotrypsin. Every time. Okay? Because it goes through this process and it ends with this genetic code that there's a protein that can decode it and make sure that this is made all the time. Okay? Now, if you thought, but it is complex, just take it this way, right? Come back to this. It's this simple, right? There's legitimately a code, and every single scientist talks about it this way. There's legitimately a digital code in our DNA that is decoded to make the proteins in our body every single second of every single day in all of our bodies. Now, take away that debate that's happened in the last 2 centuries. This is what you come across in the cell, right? Let's just say you don't have any of that context. What would you conclude? The very first thing that you would conclude is, well, what is an intelligent mind behind all this stuff, right? But we've had centuries of debate that mucked it up. You say, well, yeah, muck is the word of the day. Okay. Well, yeah, I get it, okay, but yeah, I mean, that century of debate has clouded this. It's deception, quite honestly, to see something that's so clearly true. Okay, all right.
You got 2 hours, keep going.
Are we okay? About 8 o'clock. Yeah,
Fine.
so some are like, no, please take it off. Okay, I'll go one more thing, okay? All right, so oh yeah, I have to say this. I shouldn't say this. So Richard Dawkins, do you know who Richard Dawkins is? Richard Dawkins wrote The God Delusion. He wrote The Blind Watchmaker. He is a well-known, prominent atheist, Oxford University, made billions and millions of dollars, right, selling books. Here's what he says. Biology is the study of complicated things that have the appearance of having been designed with a purpose. Is nature not speaking to Richard Dawkins? Right? Is Richard Dawkins suppressing the truth and unrighteousness? Does Richard Dawkins need our prayer? Yeah, he really does, right? I mean, it really, truly does. He says this, I have no explanation for complex biological design. All I know is that God isn't a good explanation. So we must wait and hope that somebody comes up with a better one. That's every like rush. Just to pray for it, right? I mean, that's what we're saying, right? So this is what Romans 1 is talking about. Suppresses untruth and righteousness, right? And when they come across this, they think, yeah, it looks like it's designed, but we know that can't be true because then I'd have to be accountable. It's really what they're saying. They're not saying that, but that's really what they're saying. Okay, right now, last thing here. Okay, this one will take a little bit of time. All right, so we answered this question: are there features of a biological organism that exhibit the marks of intelligence? Absolutely. I think that there are, 100%. Right? And we've got a little complicated, but I think that's right. Okay, but then the question is this: are there features of a biological organism that current neo-Darwinian theories of evolution cannot explain? That is insufficient to explain. You just think, isn't that what you just told me? Well, yeah, okay, I did. But let's go one step further in this, okay, and let's talk a little bit about something called the sampling problem. Okay, so this is really important to understand this. For and I'm going to address this from a Darwinist standpoint. Right? If you're a Darwinist, you have to think this way. Okay, biological innovation requires molecular innovation. Okay, what do I mean? Let's think about this. Okay, to get a biological innovation such as an exoskeleton, okay, you had to have the appearance of exoskeleton out of soft-bodied creatures. Okay, to get that appearance, you had to have a molecular innovation, meaning there had to be new genetic information that made enzymes that could make an exoskeleton, and that didn't exist at all in the soft-bodied animals. Okay, and the Darwinist has to say, and the evolutionist has to say, they have to be committed to say that neo-Darwinian theories can explain that innovation, and they have to demonstrate that. Okay, right. So, but there is a problem with that, and it's called the sampling problem. Now, let me put this sampling problem into this analogy for you. How many of you have ever done an escape room? Some of you. Okay, so escape rooms, really cool stuff. Our family loves escape rooms. Okay. Typically, an escape room has all these clues to get you to unlock something to get you out of the room. Okay, so there's like this big puzzle, and it takes you usually an hour to do it. Okay, and typically someone walks you into the escape room and gives you the rules and says, "Okay, find the clues," and off you go. Okay, and that's all they do. And then you spend an hour trying to find these clues that help you unlock doors and do all sorts of really fun stuff. They're really fun. Okay, if you're looking for something to do over the holidays, do an escape room. Okay, so anyway. But let's just say, though, that we walk into an escape room and the individual that's introducing it says, "I tell you what, I'm just going to give you a heads up. This box right here," they point at a box. They say, "If you unlock this box, then you will win the game." Okay, and on that box is a padlock, and on that padlock is a 4-digit sequence. Okay, with each digit has 10 possible numbers. Okay, 0 through 9. And the individual says, "You've got an hour. You can do two things. You can either seek to just go through randomly all these sequences and hope that you land on the right one, or you can look for the clues, and eventually one of the clues will tell you what the sequence is." What are you going to do? Well, if you know your probabilities, you better start looking for the clues, because you've only got an hour. Okay? There are 10,000 possible combinations in that lock. Okay? Let's say that you can sample them 1 per every second. You have 3,600 seconds to do it. You're going to only make it through a third of them. Okay? You could land on the right one, but it would be extraordinarily tedious and it would be improbable, right? Your chances, okay, are 1 in 3 that you'd be successful. Not very good. Okay? So it would be better to go look for the clues and try and find it, right? Because you've got a sampling problem. you don't have enough time to go through all the sequences. Now, if they said, you've got 12 hours, you might think it's tedious, but I'm just gonna do it. Because you'll get through all of them. At some point in time, you'll be able to get through. They give you 2 days, you're gonna get through. Fine, I'll just do it, okay? And it'd be really boring, but you don't have a sampling problem because you don't have the time, okay? All right, so what does it have to do with biology? Okay, well, remember, biological innovation requires molecular innovation, which means that we need new biological structures, we need new biological information, we need new genes, new genetic structure. We need new sequences, okay? Now, we'll come back to the sequence. 260 amino acids, okay? If we have a protein that has 260 amino acids, and there are 20 possibilities at each of the spots, we have a 20 to the 100 raised to the 160 power number of sequences that we could go through, okay? Take that into 10, right? This is 10 raised to the 338th power possible sequences of amino acids. That is a large number of sequences, but fantastically large number, okay? Now, you might think, so then are they forced to say that they have a 1 in the 10 to the 338th chance of landing on the right sequence? No, their chances are better than that, right? So understand that to get this new amino acid sequence, they have to sample through them, have to go through them, right? But understand that there are actually, because we can actually change these out, right? Remember, there's some variability, there's some tolerance for changing these out. You can land on a sequence that means something, that says something to us. More than one sequence says something to us. For instance, I could change this A to an R and probably do nothing, right? So there's 2 possible sequences that would make this protein. Okay, right? The question is, well, how rare are these specific arrangements of amino acids that will say something, that can actually make a protein? Okay, well, so there are a number of studies that have done this, okay, and I'm going to use one, 2004, Douglas Axe is the one that published this, okay, and I'm going to scale it based off of his numbers. But the odds of finding an amino acid sequence that says something to us in terms of chymotrypsin will be 1 out of every 10 to the 130th power of amino acids. Okay, now, that's a huge number, right? It's extraordinarily improbable. So do we have a sampling problem, right? Are we sitting there saying, I don't think it would be good to go through the logs, right? Because of the sampling problem. So we have to ask the question of how many sequences can you possibly get through, okay? Right, so let's give them something, okay? So let's just make these following assumptions. Let's assume that we're going to use bacteria, because bacteria they can replicate really fast, 20 minutes, every 20 minutes, okay? And let's say that we have a population of bacteria that is 10 to the 10th, okay? So 10 billion bacterial cells. Every single one of them is going to be tasked with the job of finding a new gene. Every single one of them, 10 to the 10th, okay? Now, how many times can they sample a new gene, meaning how many times can they go through a new generation? Okay, they can make up to 10,000 cells a year. One cell can make from it 10,000 separate cells, okay? So you can then in this population of bacteria, they can make or they can sample, 100 trillion, 10 to the 10th times 10 to the 4th, 100 trillion different genetic and amino acid sequences. Okay, 100 trillion, that's a lot in 1 year. Let's give them 5 billion years to do it. Some of you say I'm not comfortable with that number at all. I'm not saying that that's the number. I'm just saying that we're just going to give them that. Not only that, we're going to give them a cell. We're going to give them all the stuff that they need to find the right sequence. Okay, are they going to be able to sample enough? Well, here's our conclusion. Given them those assumptions, drastic assumptions, okay, they can only sample 5 times 10 to the 23rd sequences, which is like, over 100 orders of magnitude smaller than what they need to do. They have legitimately a sampling problem. Okay, they can't do it. It can't be done, right? So they will say, you know, you should go even further into this. Okay, scientists estimate that again using their numbers. Okay, in 3.8 billion years, that's how long they think that biological organisms have been around. There's been 10 to the 40th different organisms that have lived on the face of the planet. Okay, fine, whatever. I mean, and that would be, you know, biology, you know, that would be bacterial cells all the way up to us. Okay, there's only been 10 to the 40th. Right? We'll just say that every single one of those can sample a new genetic sequence. Okay, to find this, we're still 10 to the 40th is nowhere near 10 to the 130th sequences that they need to get through. They have the sampling problem. I mean, and so again, our question, come back here. Are there features of biological organisms that current Darwinian theories of evolution cannot explain? Yeah, there are. I think there are. Right? The rarity of sequences of proteins, right, coming to this number, presents a sampling problem for evolution. Even with their drastically large numbers, okay, and the years that I think probably most people in this room, if not all people in this room, would disagree that that's how long things have been around. Even giving them that, they're not going to make it. Okay, so yeah, so that's it. And you think, oh, well, that wasn't too bad. It wasn't 3 hours. It was an hour and 10 minutes. But Rob took some of my time.